Hello everyone. In our today's class, I am going to teach you the complement system, which is very important part of immunology. So this is unit 4 of the syllabus of core paper immunology, and in the this unit we are going to study about the complement system which is present in our body. Now the complement system consists of various proteins which we call as the complement proteins. Now what is the main function of these complement proteins which are present in the complement system? As the name suggests, complement means to aid as well as to support. So it is basically supporting the antibodies and the antigen presenting cells, which are already acting as a defense mechanisms in our body. So all the antibodies, all the antigen presenting cells which are present in our body are tackling the pathogens and tackling the foreign antigens. Similarly, the complement system has to also tackle these pathogens, these foreign antigens, and also in turn aid and assist these antibodies and the antigen presenting cells. So the discovery of complement system is actually a major effector of the humoral branch of immune system. There are two types of immunities we know, humoral and cell mediated. So from humoral branch we come to know about, and it is the main contributor of the humoral branch. So the main discovery of complement system was done by Jules Bordet. Now Jules Bordet was working on the sheep's antiserum, and here the sheep's antiserum was causing the lysis of the bacteria Vibrio cholerae. When the antiserum was heated, the antiserum became ineffective, and then afterwards it was not able to lyse the bacteria. But as soon as new antiserum was added into the older antiserum or the heat killed antiserum, but the antibodies were not added, that time also the antiserum was able to lyse the bacteria. That means apart from antibodies, there are certain other molecules also which are present in the antisera which are able to lyse and kill the bacteria. So those other molecules which are able to kill the bacteria are only the part of complement system, which are popularly referred to as the complement proteins. Moving on further, we have to talk about the functions of the complement. So functions of complement are many. Firstly, you should know that there are approximately 30 complement proteins. Some of them are soluble and some of them are cell bound proteins. When we talk about the function of the complement proteins, here are the functions. The first important function is the lysis of cells, bacteria and viruses, as we discussed above. Secondly, it does opsonization. Opsonization is a phenomenon wherein these complement proteins actually try to encircle and cover the entire bacteria. When it covers the bacteria, it is actually helping these antigen presenting cells, or actually these phagocytic cells, to engulf the bacteria. So opsonization you can relate with topping. A simple bread cannot be eaten directly, but when we apply a sauce on it, when we apply some veggies on it, we can eat it. So the function of sauce and veggies here is done by the complement proteins. So complement proteins are helping in the opsonization. Then it is helping to dilute the immune complexes which are stored in a particular part of the body. Now this is very important. There are certain cases wherein in our body some of the antigen antibody complexes get stored. Now these stored complexes are pure danger to our body, because whenever these complexes are stored, the body's immunity will be triggered again and again, and they will try to attack these immune complexes. In that condition, when again and again the same spot is triggered, and when again and again the same spot is targeted, the nearby surrounding normal cells also get affected. That is the condition of autoimmune disorder. So to avoid autoimmune disorder, the immune complexes have to be removed. These removing of immune complexes is facilitated by the complement proteins. So that is important, the clearance of the immune complexes. So the here is the antigen antibody complex, which then taken by the and opsonized by the complement proteins and finally engulfed by the pathogens. And last but not the least, these complement proteins also help to elicit the Inflammatory response. There are mast cells in our body. This is a particular mast cell. The mast cell in our body is actually responsible for release of certain chemicals. For example, when we talk about leukotrienes, for example, bradykinins, as well as prostaglandins. So the release of prostaglandins, the release of leukotrienes, and the release of bradykinins is done by these complement proteins, because these complement proteins trigger these mast cells, trigger these basophils, and trigger these other such kind of cells to release these components, and these components later cause the inflammation. So there are two important functions of complement proteins. First very important function is opsonization, so that the particular bacteria or pathogen can be engulfed by the phagocyte. Secondly, the activation of the inflammatory response. So these are the functions of complement system and complement proteins. Now we have to talk about the details of complement proteins. Now complement proteins, as I told you, there are 30 such soluble and membrane bound complement proteins, and they are designated by C1 to C9. So up from C1 to C9 there are, and then there are for certain letters also. For example, one of the complement protein is factor D. So I can say C1 is a complement protein, C2 is a complement protein, C stands for complement. Similarly, there are some other complement proteins like factor D. So these are also the complement parts only. Now for your kind information, C1 to C9 complement proteins and these certain factors like factor D, factor B, these all are further composed of two subparts, that is A subpart and B subpart, means two, basically the two fragments. And when these are activated, when the complement proteins are activated, they undergo cleavage to form the two subparts. For example, if I talk about C1, so C1 will undergo cleavage to form C1a and C1b. This can further trigger another complement protein to be cleaved into A and B. On a general note, please remember that out of the two parts or out of the two subparts, the A subpart is always responsible for causing inflammation. That is, it triggers these mast cells to release prostaglandins, leukotrienes, and bradykinins. Whereas the subpart B is always responsible for opsonization. But in the case of C2, it is an exception. In C2, the B is doing the inflammation part and A is causing the opsonization. In general sense, A is always smaller in size, that's why it can diffuse to other parts of body and cause the inflammation. But B is larger in size and therefore it stays there and it then causes the opsonization. The exception is therefore C2. Now we have to discuss the three major pathways of complement activation, and here is the clear-cut diagrammatic view of how these complement pathways get activated. So here we can see that these complement pathways are of three types. One of them is classical pathway, one of them is lectin-mannose pathway, and the one of them is, the last one is alternative pathway. So now we have to discuss about the three of them in detail. So starting with the classical pathway first. See, there are, if the pathogen has entered inside the body, then only the complement system will be activated in the classical pathway. So what happens? Generally, these complement proteins which belong to classical pathway, suppose I name them starting from C1. So this C1 protein will always be bound to the antibody. Now this antibody can be IgM most commonly, or it can be IgG with its subtypes 1, 2, 3, and 3 mainly. Whenever the foreign antigen or the pathogen binds to the antibody, the C1 gets activated. Now C1 itself is made up of complex which is C1q R2S2. This is what the subpart of C1 is. Now what happens? As soon as the antigen particularly attaches itself to the antibody, including IgM or IgG, the C1 gets activated and it undergoes self-cleavage. I am talking about classical pathway. In a way that the S unit becomes free. The S unit then acts on the activated C1, as I was telling you, that is basically C1, Q, R2, S2, and then also particularly S. This S subunit is responsible for activating C4. So it will cleave this C4 into C4A and C4B. The C4B will cause the opsonization, but the C4A will be removed out and it will be causing the inflammation. This C4B, we designate it like this, C4B. Now this C4B will further cause the cleavage of C2. It will break the C2 into C2A. I told you it is an exception, and B. The B will go out for causing inflammation. The A will be attached to C4. So it will be like, and now it will be a sequence of chain which will be formed. So it is C4B, then 2A. All of this is acting as, and it is coating the pathogen, it will be acting as the opsonizing agents. Now this particularly is C3 activator. This complex will break C3 into C3A and C3B. A part causing the inflammation, the B part will be causing what? Will be causing the opsonization. How? By attaching itself to this. So now it will be C, 4B, 2A, 3B. Now this is a particular complex which will further activate C5. Now this time it is activating C5. Now C5 is very important here. So C4B, 2A, 3B. Now what it will do? It will activate C5 and it will be called as C5 convertase. So now it will activate C5 and the C5 will be cleaved into C5A and C5B. Now this is very important inflammatory agent. So we also have a common name for this. Uh, Actually, I'm just forgetting the name of that particular C5A. Since it is causing the inflammation, so this inflammation is very important also, and we should know also about that. Anyways, nevertheless, we'll talk about it later on. So let us first focus on our pathway itself. Okay, so now we were talking about C5 convertase. Okay, now the C5 convertase, what it was doing was, it was actually trying to break the C5 into C5A and C5B. C5A is a major inflammatory agent, and C5B uh, is actually, now what we'll do, I'll explain you. I was just thinking about the name, and it is, I think so, somewhere it is anaphylatic agent or something like that, phylatic agent or something like that, which is causing the major inflammation. Now the C5B, now remember C5B, from here the whole of the game changes. Now C5 will activate, C5B will activate C6, C7, C8, C9, and these will together form the complex. Remember, these are not attaching themselves to this. There is a different thing. The C5B attaches with C6, C7, C8, C9 to form membrane attacking complex, popularly called MAC. This MAC is the one which is responsible for actual lysis. The bacterial lysis is actually caused by this MAC, or the membrane attacking complex, which is formed by C5B, C6, C7, C8, C9. We can commonly write it as C5B, then 6, 7, 8 and 9. So this is the membrane attacking complex. This is your classical pathway, wherein we have talked about that how the things work. Now in the next video we will talk about the alternative pathway.